Hello, everyone. My name is Dr. Sagar Thakkar. I'm actually a physician by training, but a nutrition scientist by trade, focusing on lactation, maternal, and early life sciences at Nestle Research Asia Hub in Singapore. Today, it's my privilege to talk to you about the past, present, and the future of human milk research. And first of all, I want to thank the organizers of NNI for the kind invitation and for me to share the topic that I'm very passionate about. With that, these are my disclosures. I am an employee of Nestle Research Asia Hub, focusing on human milk sciences, maternal nutrition, and early life nutrition. To this audience, it will not come as a surprise the benefits of breastfeeding. The World Health Organization has been communicating, along with other organizations such as UNICEF, American Academy of Pediatrics, and many Academy of Pediatrics of individual countries around the world, that initiation of breastfeeding should happen within the first hour of life. Breastfeeding should be on demand, that is, as often as child wants, day and night. It should be exclusive, that is, infant should only receive breast milk without any additional food or drink, including water. And the exclusivity of the breastfeeding should be for the first 6 months, continued up until 2 years or even beyond, with introduction of complementary food at the right time. All of this is actually summarized and highlighted in 10 steps to successful breastfeeding, specified in baby-friendly hospital initiatives. There are a lot of benefits of breastfeeding to both mother and the baby in short and long term, starting with mother-infant bonding. Additionally, to the baby, it can give protection against infections, dental caries, and malocclusions. It can also increase intelligence, reduce overweight and diabetes later in life. To moms, it can protect against breast cancer. Breastfeeding also increases birth spacing, conserving a lot of her own resources, and in long term can also lead to probable reduction in ovarian cancer and type 2 diabetes. So now I want to take a step back and talk specifically about milk, the component of breastfeeding. And the story of milk actually started millions of years ago. Actually, in early Pennsylvanian period, a terminology usually not associated with nutrition research, about 110 million years ago, synapsids laid eggs with parchment-like shells that were intolerant to desiccation, and therefore they did not survive. For survival, the synapsids started secreting glandular secretions to keep the eggs moist. This skin secretion, intended for the moisture and that worked as antimicrobial at the time, has evolved today into nutrient-rich milk long before mammals, taking the role of vehicles of nutrients and bioactivity to the newborns. These foundations were laid even before the dinosaurs walked and ruled the earth in Jurassic period. With so much evolution, it is no surprise that human milk is complex. Human milk contains variety of nutrients and bioactives. On this slide, you are actually seeing a non-exhaustive list, just what I could fit on one slide for transmission of the message, that it contains carbohydrates, proteins, individuals, lipids, minerals, vitamins, bacteria, cells, and other bioactive components. We have looked at human milk from quantitative aspects as well as structural aspects, but also organizational aspects. If we make a nutritional info of mother's milk, it would be very complex. In spite of that, I have attempted to take a snapshot of some of the critical, again, non-exhaustive list. And it would look something like what you're seeing on the screen right now. It would have calories, it would have water, fat in various amounts. It would contain vitamins and minerals important for growth and development, but also bioactives. Some of the ones that I'm going to talk about in my talk, so I have highlighted them here, and I will talk about them one at a time in subsequent slides. But first, I want to talk about the past. What has been known about human milk And this is the most studied topic, evolution along lactation. How does milk evolve with time or with the age of the baby? The first one is the amount of milk secreted or produced by the mom and therefore consumed by the baby. What we see in this tiny graph is that the amount of milk is low in the early period. The baby is still getting used to drinking milk. But then very rapidly, within 14 days, the amount of milk produced and consumed by the baby is approximately 800 mils a day. And this is a steady state average for many kids around the world. Energy also increases with passing time. Colostrum, which is the first 5 days of life, has low amount of calories compared to the transition milk and mature milk. A steady state is reached in mature milk, and the caloric content of human milk is around 67 to 70. Now, lipids are actually the main providers of the calories in milk. And we know that it reflects the same pattern as energy that you see in the second graph. Colostrum is low, but mature milk is high. And protein is actually the opposite of lipids. Colostrum is high in proteins such as immunoglobulins and cytokines that impart immunity to the baby in early life. The components of immune function actually reduces over time, and therefore you see reduction in total amount or total concentration of protein over period of lactation. In past, many papers have focused on the impact of mother's intake, be it dietary or supplementary intake, on the composition of mother's milk. We know that we can divide them majorly into two classes, modifiable and non-modifiable, but also some nutrients that you see at the bottom need additional research. Now, to highlight modifiable nutrients, I'll take the example of LC-PUFA, long-chain polyunsaturated fatty acids, also known as omega-3, and a key example is DHA. We know that higher the amount of maternal consumption of DHA, higher the amount present in the milk for the baby. Conversely, there are also non-modifiable nutrients like calcium. No matter how much calcium is consumed by the mother, the amount of calcium in mother's milk is steady state from day one. And this is because calcium is very important for the growth of the baby, and therefore mothers will do her own bone resorption to transmit calcium to the baby. Therefore, providing calcium to postpartum women is of prime importance. Geographical variation is also known to have a major impact. Maritime countries have higher omega-3s in their milk. On the right-hand side of this scale, you can look at countries like Philippines, Japan, Tanzania, and Malaysia that have access to freshwater and deep-sea fish have much higher concentrations of DHA in their milk as compared to the countries that you see on the left-hand side of this scale, such as Sudan and Argentina. What is also very well known is the gestational age at birth is also having an impact on mother's milk. Preterm milk is higher in true protein, but in colostrum only, and the difference could be up to 35%. And we know this from the systematic review and the meta-analysis done by Professor Fenton with Gidrovic. This was published in 2014. Other nutrients that are also important and highly relevant for preterm growth and survival, such as DHA, alpha-linolenic acid, which is the precursors of other long-chain fatty acids, and medium-chain fatty acids, are also found to be at higher concentration in preterm milk. We also know human milk has a circadian rhythm, not for lactose and protein, but for lipids. There is a peak in the middle of the day, perhaps reflecting maternal own dietary intake, but also giving us an opportunity for research lessons. We need to sample milk from all subjects in the study at the same time to avoid this important covariate in our results. We also know that there is within feed variation. If you hypothetically divide a breastfeeding session into thirds of foremilk, mid-milk, and hindmilk, we will see that the foremilk is a bit watery and the hindmilk is a bit creamy. Now, we have biochemical analysis proof that there are no differences in lactose and protein, but major differences in the amount of lipid present in the fore versus hind milk. I can go on and on about these research that has been done in past, but I want to summarize using this figure that shows that there are maternal factors that can influence the composition of human milk. There are infant factors that can also influence, but there are other factors such as time of day, geography, and stage of lactation, which is one of the most studied characteristics of human milk. Some of these factors are non-modifiable, but some are modifiable, and we can modify dietary intake of the mom if we want to increase bioactives such as DHA. We can also keep lifestyle factors in check and also improve the physical activity to have modulation, or they are amongst the factors that can modulate mother's milk. That was all about the past. Switching to present, we know that a lot of groups are trying to establish reference from mother's milk in form of databases. There are cross-continent collaboration using reference and gold standard methodologies. One of the key examples is in China. Professor Yin and Yang are trying to develop an online database of human milk composition from Chinese moms, but there are also efforts in the Occidental countries, such as the one that you see here, a study called Milk, NILQ, funded by Gates Foundation, which is conducted currently in United States, Europe, and Africa. Very importantly, current efforts are also ongoing in associating breastfeeding and the composition of mother's milk to public health and clinical outcomes, so that all efforts can be made to improve breastfeeding and our understanding of the association to the health benefits to mother's milk, from epidemiological perspective, from the mechanistic perspective, as well as what are the lifelong effects of breastfeeding and mother's milk consumption. If we turn towards future, we know that we have to continue the exploration of the structure-function relationship, but also pay attention to focus groups such as preterm and small for gestational age. We have to continue our bioactive exploration with advancement of the analytical technologies, understand temporal modeling as well as the interaction network within human milk. And single nutrient approach is not good enough. We need a multi-layer approach that integrates high quality of data along with good data collection with as much information as possible from both mother and the baby to have a great metadata set. We can also employ systems biology approach and the chronobiology approach. This topic has been very well highlighted by a recent paper from Dr. Parul Christian from Johns Hopkins. She says, along with her co-authors, that it is very important to understand milk interactions of internal and external factors, which can affect the composition. And composition will ultimately affect the infant health outcomes in both near and distant future. Computational and modeling methods should be employed to understanding of milk as a system, moving beyond just single nutrient identification and its association to health. And lastly, but not least, new field of cellular agriculture has emerged in the last few years. It has not been in the news, as you can see from this news clipping, that there are a lot of laboratories and startups around the world that has obtained funding from philanthropic groups like Bill Gates and his foundation to try and replicate growth of breast cells and cell-based human milk. So this is a very exciting field of human milk research to keep an eye out for in future. In a nutshell, I want to remember some of these take-home messages. First, sapien milk has co-evolved with mammalian species for millions of years. And the result is mother's milk rich in nutrients and bioactivity. Exclusive breastfeeding for the first 6 months of life provides benefit to both mother and the baby in short and long term. The past of human milk research has been focused on factors affecting human milk individual nutrients and how they have been characterized. Currently, all the efforts are going towards associating that component to function, that is to establish a structure-function relationship that is relevant to public health and clinical outcomes. However, future is exciting. With computational and modeling methods, we want to understand milk as a system, moving beyond just a single nutrient identification and structure-function relationship, especially to health. With that, I thank you for your attention.